So if you want to understand the flow of traffic in a massive, sprawling metropolis, you can't just look at a standard street map.
Right. A flat map tells you almost nothing about what's actually moving.
Exactly. To really know what's going on, you'd need like 3 different maps, and you'd have to be able to read them all at the exact same time.
Which is a headache just to think about.
It really is. I mean, the first map would show you exactly where the cars are driving right now in real time. The second map would show you which roads are actually paved, cleared of debris, and ready for cars, even if they're empty at the moment.
Okay, that makes sense so far.
But the third map wouldn't be flat at all. It would be this bizarre, constantly shifting origami structure, showing how the city physically folds in on itself.
Like a neighborhood on the far north side is somehow directly touching a neighborhood on the far south side.
Yes. And trying to decipher all 3 of those constantly shifting maps simultaneously is basically the immense, mind-bending challenge that scientists face when they try to understand how genes are regulated inside our cells.
It is a staggering level of complexity, honestly, because to truly understand why a cell behaves the way it does, you need that complete integrated picture.
You can't just look at the traffic.
Exactly. You have to look at the roads and the physical folding of the city, too. And that brings us to the core of our deep dive today. We are looking at a groundbreaking paper from Nature Biotechnology.
Yeah, detailing a revolutionary new tool called scHiCAR.
Right, which stands for single-cell Hi-C with assay for transposase-accessible chromatin and RNA sequencing. It's a mouthful.
It is a massive mouthful. But our mission today is to break down the mechanics of this tool step by step. We want to understand how it captures the RNA, the open chromatin, and the 3D folding of the exact same single cell simultaneously.
And, how it's using that to map everything from the mammalian brain to regenerating muscle tissue in just unprecedented detail.
So I want to spend a little time making sure we really understand those 3 maps first, because the whole genius of this new tool really hinges on them.
Let's do it.
Let's move away from the city analogy for a second and think of your DNA as a massive, intricate piece of origami that also functions as an instruction manual for building a human.
I like that. An origami instruction manual.
Right. So the first map we are trying to read is the transcriptome, the RNA. In our manual analogy, the RNA tells us exactly which pages are actively being read out loud and copied by the cell to make proteins.
That is the final output. That's the traffic from your city analogy.
Yes. But before a page can be read, it has to be accessible. So that is our second map, the epigenome.
Specifically, the open chromatin, right.
Exactly. Because DNA is normally wrapped up very tightly around proteins, like thread on a spool, just to fit inside the kinenucleus.
And when it's tightly wound like that, the cell's machinery just can't read it.
It's totally locked away. The cell has to physically unspool specific sections to access the instructions. So we measure this open chromatin to see which pages of our manual are actively bookmarked or, held open.
And those open areas are crucial. They contain these candidate cis-regulatory elements, or CCREs.
Like promoters and enhancers, right?
Exactly. Promoters are essentially the on switches sitting right next to a gene. And enhancers are, they're fascinating because they act like powerful volume dials for the gene.
But those dials can be located incredibly far away on the linear DNA strand.
Right, which is why that distance makes the third map so crucial. The 3D genome or the Hi-C data.
Because this instruction manual isn't laid out flat on a table. It is intricately folded up.
So page 10 might be physically touching page 10,000?
And in biology, we call these long-range cis interactions. Cis just meaning it's happening on the same molecule of DNA.
Right. So an enhancer on page 10,000 can reach over and turn up the volume on a promoter on page 10, but only if the 3D origami folding brings them into physical contact.
Okay, so that's the setup. We have the RNA output, the open chromatin roads, and the 3D folding.
And the absolute necessity here, and really the historical bottleneck in the entire field, is capturing all 3 of these modalities from the exact same individual cell.
Because if you just take a bulk tissue sample, you're essentially blending millions of cells into a smoothie.
Exactly. You get an average flavor, but you completely lose the specific details of any individual cell.
But wait, haven't researchers done this before? Like capturing multiple things from one cell?
Well, previous single-cell methods could capture maybe 2 out of the 3 layers, like scHiCAR or scHiCAR.
Right, or they would take separate datasets from different cells and try to use algorithms to just, computationally smash them together.
Yes, computational integration. But when you rely on guesswork without a true single-cell ground truth, You inevitably create blind spots and artifacts. You just miss the nuanced cell-to-cell differences.
But I'm still hung up on the 3D part. Don't we already have ways to look at how DNA is folded in single cells?
We do, yeah.
Like, I feel like we've seen single-cell 3D mapping before. Methods like DIPSI or LimMCA or SNM3C. Why go through the trouble of inventing an entirely new tool just to get this third map?
It really comes down to signal versus noise. Those older single-cell 3D methods you mentioned are what we call unbiased.
Meaning they just look at everything.
Exactly. They capture all DNA contacts indiscriminately, so they map out the folded connections of the deeply packed, inactive garbage DNA just as thoroughly as they map the crucial open regulatory regions.
Oh, I see. So it's mostly junk data.
Huge amounts of it.
Yeah.
Because they capture everything, you have to sequence billions of pieces of data just to find the tiny fraction of interactions that actually matter.
That sounds astronomically expensive.
It is. And even with all that sequencing, only about 6 to 10% of your data represents the long-range enhancer promoter loops you actually care about.
Wow, only 6 to 10%. That's terrible efficiency.
And on top of that, those older methods usually require you to process cells one by one in separate tubes, which just completely ruins your throughput.
Okay, so let's do a quick recap here for you listening, just to make sure we've got all these puzzle pieces locked in before we look at how this new tool actually works.
Good idea.
So bullet 1, gene regulation requires 3 things: the RNA, the open chromatin, and the 3D structure.
Bullet 2. Trying to integrate separate datasets computationally causes artifacts because it lacks a ground truth from a single cell.
And bullet 3, older single-cell 3D methods are unbiased, meaning they are inefficient, expensive, and yield very low percentages of useful data.
Perfectly summarized.
And if you're listening and wondering why scientists don't just guess how these systems fit together, I mean, have you ever tried piecing together a complex jigsaw puzzle where the pieces actually came from 3 different boxes?
It's a total mess. Consider how much easier it is when all the pieces are verified to come from the exact same box.
Right, which is exactly the problem that scHiCAR was engineered to solve.
Yes, and the workflow of scHiCAR is essentially a microscopic assembly line.
The numbers on this are staggering to me. The paper says a single researcher can complete this entire protocol in, what, 3 to 4 days?
Yep, 3 to 4 days. And it costs roughly 4 cents per cell.
Tracking 3 distinct layers of biology for 4 cents a cell is wild. How do they even label millions of individual cells without needing millions of microscopic tubes?
The secret to achieving that kind of scale is a statistical trick called combinatorial barcoding.
Walk me through that.
So it starts by taking the cells and chemically cross-linking them. This is basically like spraying the nucleus with a chemical fixative.
So it freezes that 3D origami structure exactly in place.
Exactly. It locks everything down. Then they take these frozen nuclei and put them into a standard 96-well plate.
Just a standard plastic plate with 96 little dimples in it.
Right. And into each well, they drop a special enzyme called TN5.
Oh, I love the TN5 enzyme. It's basically a tiny molecular staple gun, right?
That is a perfect way to visualize it. TN5 specifically hunts down the open, accessible chromatin, the unwound thread, and it inserts chemical tags directly into it.
And it completely ignores the tightly packed DNA.
Completely ignores it. So at the exact same time, in that same, well, they also convert the cell's fragile RNA into a more stable format called cDNA.
Okay, so we've got tagged open chromatin and we've got our RNA copies.
Right. And here is where the barcoding starts. They attach barcode 1 to both the TN5 tags and the cDNA in that specific well.
So like every cell in well number 1 gets a barcode 1 sticker.
Yes. Then they take all the cells from all 96 wells, pull them together into one big tube, mix them all up, and then randomly deal them back out into a brand new 96-well plate.
And in this second plate, they attach barcode 2.
You got it. Then they mix them all up again and deal them out into a third plate to attach barcode 3. It is just a cycle of pooling and splitting.
Okay, and the math behind that pool and split method is where the real magic lies, right? Because 96 times 96 gives you nearly 85 million unique barcode combinations.
Exactly. Statistically, every single cell that travels through those 3 rounds of mixing and tagging is going to end up with a completely unique 3-part molecular address.
Okay, so we have a permanent address for every cell. But this brings up a huge logistical problem. Well, you now have the RNA copies and the massive 3D folded DNA sitting inside the exact same tiny nucleus, sharing the same barcode.
Right.
If you put that entire nucleus into a sequencing machine as is, the signals would just be an unreadable mess, wouldn't they? How on earth do they physically separate the RNA from the DNA without losing track of which cell they belong to?
The solution they came up with is wonderfully physical and honestly pretty low tech.
Really? How so?
They use a 0.5% SDS treatment. SDS is just a mild detergent. It's very similar to what's in your dish soap.
Wait, dish soap?
Basically, yeah.
Okay.
By applying exactly this concentration of detergent, they gently permeabilize the nuclear membrane. They just poke microscopic holes in it.
Oh, I see where this is going.
Right, because the RNA copies the cDNA are relatively small linear molecules, so they simply leak out through those microscopic holes into the liquid floating above the cells.
Meanwhile, the 3D genome is this massive, tangled, chemically frozen hairball of DNA. It's way too big to fit through those tiny holes.
Exactly, so it stays safely anchored at the bottom of the tube in a solid pellet.
That is brilliantly elegant. You literally just take a pipette, siphon off the liquid at the top to get your pure RNA library, and keep the heavy pellet at the bottom for your 3D DNA library.
It's beautiful in its simplicity.
It is. But I have to push back on the next step.
Okay, let's hear it.
You have this heavy pellet of frozen 3D DNA. To map the connections, the protocol says they chop the DNA up with molecular scissors, like a 4-base cutter called CVQI.
That's right.
And then they use an enzyme to glue the loose ends back together in situ.
Hold on. If you're chopping up the DNA and gluing it back together inside a tiny, chaotic nucleus, how do you know you aren't just accidentally gluing together random pieces of garbage DNA that just happen to be floating near each other?
That is a great question, and it's the exact reason they cross-link the cells at the very beginning.
Oh, the chemical freeze.
Yes, because the proteins holding the DNA together are chemically frozen. When you cut the DNA strand, the pieces don't just float away. They are held rigidly in place in 3D space, so they can't mingle.
Right, so when you add the gluing enzyme, it can only reach and glue together DNA ends that are physically touching each other in that folded origami structure.
Okay, that makes sense. So you are genuinely capturing pieces of DNA that are physically interacting, even if they are millions of base pairs apart on the linear sequence.
Exactly.
But how does scHiCAR filter out the noise? You mentioned earlier that older methods waste money sequencing all the tightly packed, inactive DNA. How does this new tool only pull out the critical loops happening at the open chromatin?
This is where we bring it all back to that TN5 staple gun from the very first step.
The one that only targets open chromatin.
Right. So after they cut and glue the 3D folded DNA together, they use another set of molecular scissors, an enzyme called NLII, to cut it again.
Okay.
And this time, they force those glued fragments to self-ligate into tiny, circular pieces of DNA.
Circles. Why circles?
Because finally, they use PCR primers, which act like molecular homing beacons. And these primers are specifically designed to only recognize and amplify sequences that contain that original TN5 tag.
Oh, that's incredibly clever. It's like a GPS tracker.
That's exactly what it is.
The primers completely ignore the billions of random closed-off DNA loops. They only amplify the 3D connections that are anchored to an open piece of chromatin.
And that targeted enrichment changes everything. It boosts the capture of crucial long-range cis interactions to 48%.
Compared to the measly 6 to 10% seen in older methods.
Right. It turns a blunt instrument into a highly precise surgical tool.
Amazing. Okay, let's do another quick recap and check our mental model here.
Sounds good.
So bullet 1, we use combinatorial barcoding, which is pooling and splitting cells, to label millions of them for just pennies.
Bullet 2. We use a mild detergent, SDS, to physically separate the RNA from the folded DNA. The tiny RNA leaks out. The massive DNA stays in the pellet.
And bullet 3, we chop up the 3D genome, glue it together, and use the TN5 tags as a GPS tracker to strictly pull out only the active functional loops.
Perfect.
So for you listening, can you visualize that moment? The mild detergent releases the RNA into the liquid while the folded DNA remains anchored. It's just such a cool physical solution to a complex chemical problem.
It really is. But, you know, an impressive engine doesn't mean much until you take it out on the road.
Right. They proved this worked on human and mouse cell lines first, with like 100% classification accuracy.
Yeah.
But then they needed a massive real-world test.
So they turned to the mammalian brain.
They mapped 1.62 million mouse frontal cortex cells. That number is just hard to wrap your head around.
To put that into perspective, this single scHiCAR experiment generated a data set larger than the entire highly celebrated BICCN whole mouse brain data set for 3D genomics.
Wow. And what did they find in all those cells?
Well, by analyzing those 1.62 million cells, they clearly identified 22 distinct major brain cell types.
Like specific neurons and support cells.
Exactly. They could map out highly specific populations, like p-valve inhibitory neurons or support cells like astrocytes, and they could see exactly how the transcriptome, the open chromatin, and the 3D folding differed in each unique cell type.
But reading the paper, there's a massive computational wall they had to climb here.
Yes, the data sparsity.
Right. Even with 1.6 million single cells, the 3D contact data for any one individual cell is incredibly sparse.
It is. You might only capture a few thousand physical contacts out of billions of possibilities.
So the map is basically just a few blurry dots. How do you look at that incredibly sparse data and confidently claim, yes, this specific enhancer loops exactly to this specific promoter?
That sparsity is the fundamental bottleneck of all single-cell 3D genomics. You have a blurry map. To solve this, the researchers built an artificial intelligence algorithm called scDeepLUCIA.
scDeepLUCIA.
It's a specialized deep learning loop caller, basically a neural network specifically designed to find these hidden loops.
But how does an AI find loops in a map that's mostly empty space?
Okay, think of the blurry 3D data like a highly degraded satellite photo of a city at night. You can barely make out any features.
Okay, I'm picturing it, just a few fuzzy pixels of light.
Right. But because this is scHiCAR, you also have a second map from the exact same cells, the open chromatin map.
The Rhodes map from earlier.
Exactly. Think of the open chromatin as a crystal clear map of where all the streetlights are turned on.
Oh, I see.
The AI is trained to understand the biological rules of folding. It knows that major highways, the 3D loops, usually connect clusters of streetlights, which are the open chromatin.
So it takes the clear streetlight map, superimposes it over the blurry satellite photo, and constantly draws in the precise neural pathways and highways.
You've got it. It
Yeah.
uses the 1D open chromatin data to resolve the blurry 3D contact data into high resolution.
And it doesn't need to be deeply trained on bulk data from the exact same tissue beforehand, right? It just learns the rules of de novo prediction and applies them.
Right, and they proved it actually works in the real world. They used this AI to look for functional enhancers, and it outperformed existing gold standard models like the ABC model.
Oh, they used the Vista enhancer database to check it, didn't they?
They did. For instance, the AI pinpointed a specific enhancer called MM1C79. The scHiCAR data predicted this enhancer was looping across a massive distance, nearly 500,000 base pairs.
Wow.
Right, looping all the way over to activate a specific gene called chromatin inside a highly specialized type of neuron, the L23IT1 cells in the upper cortex.
And when they cross-referenced this prediction with the Vista database of experimentally validated biology,
The database confirmed it. The enhancer was real, and it was active in the exact brain region the AI predicted. It literally found the exact needle in a 1.6 million cell haystack.
the precision is astonishing.
It represents a total paradigm shift in computational biology.
Okay, let's pause for recap number 3.
Let's hear it.
Bullet 1. scHiCAR successfully mapped 1.62 million mouse brain cells across 22 distinct cell types.
Bullet 2. To overcome single-cell data sparsity, they used an AI algorithm called scDeepLUCIA, which uses 1D open chromatin to predict high-res 3D loops.
And bullet 3. This method is highly accurate at predicting real, functional enhancer gene pairs, like finding that MM1679 enhancer.
Spot on.
And for you listening, just think about how incredible it is that an AI can look at a blurry, sparse 3D map of a single cell, cross-reference it with open chromatin data, and accurately predict the exact molecular loops connecting a distant enhancer to its target gene. It's like magic.
It feels like magic, but it's just really good math. But, mapping the static brain, as complex as it is only half the story here.
Right, because the true test of gene regulation is watching it respond to stress. What happens when a tissue isn't static, but is actively tearing itself apart and rebuilding?
Exactly. So the researchers transitioned from the brain to skeletal muscle.
And this is where the timing of all these maps gets really wild.
They used a mouse model where they inject barium chloride, or BaCl2, into the muscle.
Which causes a controlled acute injury to the tissue. Right.
Right. And this injury model allows them to track exactly how the muscle repairs itself over time. They tracked over 40,000 cells across 3 specific time points. Day 0, which is uninjured, Day 5, and Day 7 post-injury.
And they were specifically looking at muscle stem cells, the PAX7 positive cells.
Yes. Normally, these stem cells are quiescent. They are completely asleep. But the injury serves as a massive alarm bell.
So they wake up, multiply, and transition through several stages, like becoming myoblasts and myocytes, to eventually repair the damage.
Exactly. The researchers mapped these cells along a developmental timeline, which we call a pseudo-time trajectory, to watch the genes turn on step by step.
And this is where my entire mental model of biology got flipped upside down when reading this.
Oh, yeah. How so?
Well, I always pictured gene activation as a perfectly synchronized event, like flipping a light switch. I assume the 3D origami genome unfolds, the chromatin opens up so the enzymes can bind, and the RNA is immediately transcribed all in one fluid simultaneous motion.
That is exactly the assumption that the scHiCAR data dismantles. By tracking all three modalities in the same cell along that timeline, they found that the timing is entirely gene-specific.
Meaning they don't all happen at once.
Right, and crucially, they discovered that the 3D structural folding of the genome can actually happen before the RNA transcription begins.
Wait, really? The genome physically solds to connect the enhancer and the promoter before the gene is even turned on?
Yes. They zoomed in on crucial genes required for muscle development, like MYOG and MY1, and they could literally watch the timeline.
So what did it look like?
The genome reorganized its 3D contacts, forming the intricate loop between the volume dial and the on switch, well before the RNA machinery actually started churning out copies of the gene.
That is, wow. It's the biological equivalent of building the elaborate scaffolding and supply lines around a factory floor weeks before the factory actually opens for production.
That's a great analogy. It implies a level of anticipation in the cell that is just wild.
Okay, final recap here to pull all these threads together.
Go for it.
Bullet 1. scHiCAR works on dynamic, challenging tissues, not just static ones, which they proved by tracking regenerating skeletal muscle over 7 days.
Bullet 2. By tracking muscle stem cells, it proved that gene activation is not a single synchronized switch. The timing varies by gene.
And bullet 3, the massive revelation. The physical 3D origami folding of your DNA can act as a precursor, happening before the actual RNA output begins.
Exactly.
So if you are listening to this, I want to leave you with a question that scientists are scrambling to answer right now. If the 3D folding of the genome happens before a gene is actually turned on and transcribed into RNA, what is the director in the cell that is telling the genome to fold in preparation?
Right. What is the signal? Unlocking that hidden director will likely define the next decade of genomic research. It proves that the genome isn't just a passive storage drive. It has structural memory and anticipation built into its very folds.
It's almost like it's thinking ahead.
It is. And if we extrapolate that finding into the clinic, the implications are profound. Well, if we know that 3D folding can precede RNA expression, we might be able to use this technology as a structural early warning system for disease.
Oh, wow.
Imagine taking a biopsy from a patient and using scHiCAR to map the 3D folding of their cells. We might be able to spot the dangerous abnormal genome folding that leads to cancer before the actual malignant oncogene RNA is ever transcribed.
So we could see the cancer scaffolding being built before the tumor even really exists.
Exactly. We could catch it at the structural folding stage before the dangerous output starts.
A structural early warning system hidden in the folds of our DNA. That is a truly incredible thought to end on. Thank you for joining us on this deep dive. Keep questioning how things work and keep wondering about those 3 shifting maps operating right now in real time inside every single one of your cells.